Now, what happens up here that we have to look at is we're going to digress a minute and look at chromosomes. This is a chart of human chromosomes that have been taken out of a cell and using very special separation techniques and microdissection have been lined up in pairs from the biggest to the smallest. They can be matched manually by the way they look. If you notice, chromosomes have two sets of arms. They have what are called the P arms, or the petite, the small arms, and then they have the long arms down here. And there are 23 pairs. When I was in high school, we thought there were 24 pairs because the first investigators actually counted the same chromosome twice. And nobody challenged it, and this went on. It's in textbooks, and I grew up thinking there was a total of 48 chromosomes and not 46 from the 23 pairs. If you'll notice, the ones that are different is the number 23, which has an X and a Y. These are the sex chromosomes. There's virtually no information on the Y chromosome, but the X chromosome and the Y are called the sex chromosomes. We'll talk about those again later. Of other interest here is chromosome 21. When there are 3 of these by a genetic mistake, you have something called trisomy 21, which we know as Down syndrome. It used to be called Mongolism. It's a combination of physical and mental retardation, very, very common, and common in aged eggs and aging sperm as well. So the older parents tend to have children with Down syndrome. These chromosomes this is the diploid number. We have 2 of each. So what happens when we go in both the female and male reproductive system? This slide shows the progress and the divisions that take place. It's upside down relative to the slide I showed you. So here's the outside, the basement membrane of the seminiferous tubules. This is the superficial side. We have daughter cells. Here is our basic stem cell with the 2N number, the 46 chromosomes. It's a spermatogonium, and it will divide with something called mitosis, which means the whole pair of chromosomes will divide and will have the complete number still. There's no loss of chromosomal material. One of those will become a daughter cell, so we keep our number of stem cells. And the other one will undergo differentiation to be pushed away from the basement membrane and differentiate into that primary spermatocyte, which we saw before. It still has 2N. DNA replication and crossing over occurs here. If we did not have crossing over is when the arms of the chromosome exchange DNA. I give you part of yours and you take part of mine, and now we have a new strand and combination of traits. If that didn't happen, we would tend to be exactly like our parents. There'd be no genetic shuffling of traits and recombining. Nature found this to be, or evolution found this to be, very beneficial if we could shuffle the deck every now and then. So instead of having a repeat of every generation when we get the similar parents, in every division here that occurs between the primary spermatocyte before we get into the secondary spermatocyte, and the same thing occurring in the eggs, the cards are shuffled. And the way they're shuffled is by random crossing over and exchange of similar chromosomes. So number 21 would exchange with 21. They wouldn't exchange with another chromosome. So they would be exchanging the same locations of their genes, but they might have different attributes. One might have brown eye, blue eye, different things of all our characteristics would be constantly shuffled, millions and millions of times. So we get this this array of different traits, and then evolution is going to work on those different traits, see which ones are better, so we have selection of the fittest. At this point, a type of division which is no longer called mitosis, but rather meiosis, is reduction division. And in reduction division, you only carry away the one of each of the pairs of chromosomes. So now each of these guys has only half the complement. They each have 1 through 23 a single copy of each in normal circumstances. Finally, they become the spermatotids, each with a single number, and finally we get the sperm cell. Now, the germ cells in both the male and female are carrying half the number, so when they join again, we'll be back to the normal number. We don't want to have more than the normal number of chromosome or DNA material. It always leads to errors. Nothing good comes of it. We do have cases where there's 3 X chromosomes and 1 Y or no Y. Some of these are called super females, but they're really not. They are actually usually have great many problems in all their systems. So reshuffling is important for natural selection. Also maintaining the diploid number in the adult and the haploid or half number in the germ cells, which are the only cells that have that, is very important in nature, and keeping it that way so that we don't have any bad traits coming through because of too much or too little